We often think of our genes as a fixed blueprint dictating our traits and predispositions, but what if this blueprint is not set in stone? What if our experiences and environment could actually influence how our genes are expressed? This is the fascinating realm of epigenetics. Epigenetics is the study of changes in gene expression that occur without alterations to the underlying DNA sequence. It's like having a dimmer switch that can turn the volume of a gene up or down without changing the song itself. This revolutionary field is transforming our understanding of inheritance, health, and even the very nature of life itself. Imagine our DNA as a vast library of genes, each containing instructions for building and maintaining our bodies. Epigenetics acts as the librarian, determining which books are accessible and how frequently they are read. This intricate system of regulation ensures that the right genes are expressed at the right time in the right cells. It's a dynamic and responsive process, allowing us to adapt to our surroundings and experiences throughout our lives. Epigenetics is revealing that we are not merely passive recipients of our genetic inheritance, but active participants in shaping our own biological destinies. The implications of epigenetics are far-reaching, touching upon everything from disease susceptibility to personality traits. By understanding the mechanisms that govern gene expression, we gain valuable insights into how our environment and lifestyle choices can impact our health and well-being. This knowledge empowers us to make informed decisions that can positively influence our own lives and the lives of future generations. The study of epigenetics is not just about understanding our past, but also about shaping our future. Epigenetic changes occur through a variety of molecular mechanisms, with DNA methylation and histone modification being two of the most well-studied. These processes act like tiny switches, flipping genes on or off without altering the underlying DNA sequence. They are essential for normal development and play a crucial role in how our bodies respond to environmental cues. Imagine a conductor leading an orchestra using subtle hand gestures to control the volume and tempo of the music. Similarly, DNA methylation and histone modification fine-tune the expression of our genes, orchestrating the complex symphony of life. DNA methylation involves the attachment of a methyl group, a simple chemical tag, to specific regions of the DNA molecule. This typically acts as a silencing mark, preventing the gene from being transcribed into RNA and subsequently translated into protein. Think of it like placing a bookmark on a page, making it harder to read. On the other hand, histone modification involves chemical changes to histones, the proteins around which our DNA is tightly coiled. These modifications can either loosen or tighten the DNA packaging, making the genes more or less accessible for transcription. The interplay between DNA methylation, histone modification, and other epigenetic mechanisms creates a complex regulatory landscape that governs gene expression. These processes are influenced by a multitude of factors, including our diet, exposure to toxins, stress levels, and even social interactions. This dynamic interplay between our genes and our environment highlights the remarkable plasticity of our biology. It underscores the fact that we are not simply hardwired by our genes, but rather shaped by the intricate dance between nature and nurture. Identical twins, sharing the same DNA sequence, offer a fascinating window into the power of epigenetics. While these twins start life with virtually identical genetic blueprints, their epigenomes, the collection of epigenetic marks on their DNA, can diverge significantly over time. This explains why identical twins, despite their genetic similarities, can exhibit striking differences in their physical characteristics, disease susceptibility, and even personality traits. Studies on identical twins have revealed that epigenetic differences accumulate as twins age, particularly if they experience different environments or lifestyles. For instance, twins who grow up in different households, have different diets, or experience varying levels of stress may exhibit distinct epigenetic profiles later in life. These epigenetic variations can influence a wide range of traits, from their height and weight to their risk of developing diseases like cancer or heart disease. The case of identical twins highlights the remarkable plasticity of our epigenomes and their susceptibility to environmental influences. It underscores the fact that our genes do not dictate our destiny, but rather provide a framework upon which our experiences and choices can sculpt our lives. The study of twins provides compelling evidence for the significant role of epigenetics in shaping who we are, independent of our genetic blueprint. It paints a picture of human development where nature and nurture are not separate forces, but rather engaged in a continuous, intricate dance. The emerging field of social epigenetics explores how social factors such as socioeconomic status, discrimination, and early life adversity can leave lasting marks on our epigenomes, influencing our health and well-being. This research has profound implications for understanding health disparities and promoting health equity. It sheds light on how social injustices can become biologically embedded, perpetuating health inequalities across generations. Studies have shown that individuals from disadvantaged backgrounds often exhibit distinct epigenetic profiles compared to their more advantaged counterparts. These differences can manifest in a higher risk of chronic diseases such as heart disease, diabetes, and certain types of cancer. For example, children who experience chronic stress early in life, such as poverty or abuse, may undergo epigenetic changes that affect their stress response systems, making them more vulnerable to stress-related disorders later in life. Recognizing the role of social factors in shaping our epigenomes challenges traditional notions of health and disease. It compels us to look beyond individual behaviors and genetic predispositions to address the social and environmental factors that contribute to health inequalities. By understanding the epigenetic mechanisms that underpin these disparities, we can develop targeted interventions and policies aimed at creating a more just and equitable society. Epigenetics provides a powerful lens through which to view and address health disparities, advocating for a more holistic and socially conscious approach to healthcare. One of the most intriguing aspects of epigenetics is the potential for these changes to be passed down from one generation to the next. This means that our experiences and lifestyle choices today could have implications for the health and well-being of our children and grandchildren. While the idea of inheriting acquired traits was once dismissed, epigenetic inheritance is now recognized as a significant factor in understanding the transmission of health and disease across generations. Research has shown that environmental exposures such as diet, stress, and exposure to toxins can induce epigenetic changes in parents that are then passed on to their offspring. For example, studies have shown that pregnant women who experience famine or severe stress during pregnancy may have children with an increased risk of developing metabolic disorders and mental health issues later in life. The concept of epigenetic inheritance has profound ethical and societal implications. It highlights the interconnectedness of generations and underscores the responsibility we have to create a healthier and more just world for ourselves and for future generations. Understanding the mechanisms of epigenetic inheritance can lead to the development of interventions that mitigate the negative effects of environmental exposures and break the cycle of transgenerational disease transmission. Epigenetics is revolutionizing our understanding of human health and disease, revealing the dynamic interplay between our genes, our environment, and our life experiences. It is transforming our perception of inheritance, demonstrating that we are not simply passive recipients of our genetic blueprint, but active participants in shaping our own biological destinies and those of future generations. This knowledge brings with it both immense promise and profound responsibility. The promise of epigenetics lies in its potential to develop new diagnostic tools, therapeutic interventions, and public health strategies. By understanding the epigenetic mechanisms that underpin disease, we can develop more targeted and effective treatments. Furthermore, by identifying environmental factors that influence gene expression, we can design interventions and policies that promote health and prevent disease. Epigenetics offers a new frontier in personalized medicine, allowing us to tailor treatments based on an individual's unique epigenetic profile. However, the power of epigenetics also raises ethical considerations. As we unravel the complexities of gene regulation, we must grapple with the potential for misuse of this knowledge. Issues such as genetic privacy, discrimination based on epigenetic information, and the potential for epigenetic engineering require careful consideration and ethical guidelines. The study of epigenetics is a testament to the remarkable adaptability and resilience of life. It underscores the interconnectedness of all living things and the profound influence our environment and choices have on our health and well-being. As we delve deeper into the epigenetic landscape, we embark on a journey of discovery that promises to transform our understanding of ourselves and the world around us. Ultimately, epigenetics empowers us to become active architects of our own health and contribute to a healthier and more equitable future for all.